Hello everyone, welcome back. In continuation with my previous video, in this video let us discuss about other two cloning vectors, that is Cosmids and Phage vectors. The target host organism for both these vectors is bacteria. Now first let us discuss about Cosmids. These Cosmids are nothing but the plasmids with lambda phage cos sites. These cos sites are nothing but the cohesive ends. We already discussed about the blunt ends and the cohesive ends that are produced after the digestion of DNA with the restriction enzymes, right? So for example, if we consider it as a double-stranded DNA molecule, when we digest this DNA molecule with restriction enzymes, some of the restriction enzymes will cut a single-stranded DNA at both the ends, leaving overhangs. Okay, see here, this produced ends are nothing but the sticky ends, or we can also call them as cohesive ends. Whereas in some cases, some of the restriction enzymes will make a double-stranded DNA cut, leaving blunt ends. Hope you are clear with the difference between the blunt end and the sticky end. If there is a double-stranded cut, then it will give rise to a blunt end. But if there is a single-stranded DNA cut at both the ends, leaving overhangs, that results in the cohesive ends or sticky ends. Right? Due to the presence of this cos sites or cohesive ends, it is very easy to pack them into phage heads. So these Cosmids will have cos sites at both the ends. And in order to make a recombinant DNA molecule, we need to introduce this foreign gene into a vector, right? This DNA of our interest will be inserted in between these cos sites, so that it will be easily packaged into a phage head, which we call it as in vitro packaging. They can also exist as plasmids within the bacterial host. By using this Cosmids, we can clone DNA fragments of up to 50 kilobases. These Cosmids were first developed by Barbara and John Collins in the year 1978. Look at the picture carefully. This is the Cosmid. Cosmids are nothing but the plasmids which will have cos sites or cohesive ends, right? They will have origin of replication site, cleavage site and a marker gene which codes for ampicillin resistance. If we want to make a recombinant DNA molecule by introducing this foreign DNA into this Cosmid vector, we need to open the vector, right? So for that reason, it should possess cleavage site, where the restriction enzymes are going to digest the DNA exactly at this location and they are going to open the vector. Then we are going to introduce this foreign DNA into this vector to make a recombinant DNA molecule. Now this foreign DNA is introduced into this vector with the help of an enzyme called T4DNA ligase. First, this Cosmid is linearized with the help of restriction enzymes. See here, it is in circular form. Now it is linearized by treating it with the restriction endonucleases. At the same time, this foreign DNA is also partially digested with the help of restriction enzymes, which will give rise to a DNA fragments. Now this DNA fragment is to be inserted into this Cosmid vector to make a recombinant DNA molecule, right? And the size of this Cosmid is 4 to 6 kilobases. Smaller the Cosmid, we can introduce larger the DNA fragment. So with the help of this Cosmids, we can introduce DNA fragment of about 45 to 50 kilobases. Ligation of mixed DNA preparations results in the formation of concatemers. And the use of concatemers in high concentration is really favorable. So now we need to prepare a recombinant DNA molecule, right? See here, this is the foreign DNA and this is the Cosmid vector. With the help of an enzyme called T4DNA ligase, we are going to insert this foreign DNA in between the cos sites, making a recombinant DNA molecule. See here, this is the foreign DNA that got incorporated in between cos sites. Due to the presence of this cos sites, which we also call them as cohesive ends, it can be easily packaged into phage heads, and that process is called as in vitro packaging. Now these phages are ready to infect the bacteria. Here the bacteria E. coli is the host organism, right? So after the infection, now we are going to screen for the presence of transformed cells with the help of antibiotic resistant marker genes. Coming to the other important cloning vector, that is phage vectors. Bacteriophages or phage vectors are one of the important and commonly used cloning vectors in genetic engineering. Both single-stranded and double-stranded phage vectors are being used. And these phage vectors are really helpful compared to the plasmid vectors, where they can accommodate larger DNA fragments, which is not possible with plasmid vectors. These phage vectors can carry larger DNA fragments of up to 24.6 kb in length. And these phage vectors are the most suitable cloning vehicles which are used to clone genomic eukaryotic DNA. And one more important use with this phage vectors is that in vitro packaging is possible. You all know very well about a bacteriophage, right? These bacteriophages are nothing but the viruses which infect the bacteria. So here the bacteria is the host organism, right? Whenever this bacteriophage comes in contact with the bacteria, so this genetic material will be ejected into the bacteria. If we look into the life cycle of a bacteriophage, it will have lytic cycle and lysogenic cycle, right? In case of lytic cycle, it is going to destroy the host organism. All the produced new viral particles will come out of the cell after the lysis of the bacterial cell. Whereas coming to the lysogenic cycle, it is going to integrate with the host DNA. In this way, DNA is successfully delivered into host organism. If we look into the genome of a bacteriophage, 1/3 of the genome is non-essential. So we can replace this portion with foreign DNA. Look at the picture carefully. This is the lambda phage DNA which is treated with restriction enzymes. So here the non-essential portion is removed and it is replaced with the foreign DNA. See here, this DNA is also treated with the restriction enzymes, giving rise to DNA fragments. Out of this DNA fragments, we are especially interested in this gene. So we can call this gene as DNA of interest. Now this gene is to be introduced into this vector to make a recombinant DNA molecule. For that reason, we take help of an enzyme called DNA ligase, which is going to join this foreign DNA with this vector. Now it is a recombinant DNA molecule which can be reintroduced back into a phage head. See here, it is possessing cos sites or cohesive ends, which is easy to package them into a phage head by a process called in vitro packaging. Now these phages are ready to infect the host organism. After the process of infection, now we are going to screen for the presence of transformed cells. This is all about the cosmids and phage vectors. Soon I will come up with my next video. Thank you so much.